Recording. Okay. Hello, everybody. Welcome to another session of our seminar. It's a real pleasure for us to welcome today Professor Martin Falcke, if I pronounced that correctly. Sorry if I didn't.
Yes, you did.
Who's in Berlin. Martin was initially in industry and then came to science, which is interesting for the students here, I guess. Got a PhD in 1995 at the Technische Universität Berlin in theoretical physics. Then did a postdoc in chemistry in the University of Virginia, at which time the transition to life sciences kind of started, as I gathered, and then was in San Diego at the Institute for Nonlinear Science, and then in the 2000s was at the Hahn-Meitner Institute in Berlin as a PI in theoretical physics, and currently Professor Falcke is one of the leading people working in several areas, including calcium signaling and others, and he is currently a PI at the Max Delbrück Center for Molecular Medicine in Berlin and a professor for biological physics at the Department of Physics, Humboldt University, Berlin. So over to you, Matt.
Thank you for the kind introduction. The Quantitative Life Science Consortium at McGill is a pretty impressive lineup of people. And together with CAMBAM, I think this is a great biotope for quantitative life sciences. So it's a real pleasure for me to present some of our work in this seminar, and I'm really grateful for this invitation. So the work I will present has been a collaboration between the experimental lab of Joachim Redlers in Munich, and there it has been done by Christoph Schreiber, Johannes Hein, Santiago Kuhl, and Esra Ekin Teleboglu and Gerlinde Schwake. And the theory has been done by Behnam Amiri and Rainer Marcel Eberhard in my lab. So here you see two examples of cells moving on two-dimensional substrates. And you can see here with the B16 cell, the cell body, and then this huge flat protrusion. And similar, here with the fish keratocyte, you also can see the cell body, and again, this huge flat protrusion. So the height ratio between the protrusion and the cell body is similar to the height ratio between the shield and the cap of a base cap. So these are really large flat structures, about 100 to 200 nanometers in height. If we remove the cell membrane from the cell in the protrusion, we see a dense network of actin filaments. And here you see an electron tomography tomogram of such a network. The front and the rear part are magnified in C and D here. And here at the front, very front of the network, you see filament tips sticking out of the network, and this is where polymerization of these filaments goes on and generates a force and driving motion against the leading edge membrane of the cell. And in the rear, these filaments depolymerize and bundle up. The polymerization of these actin filaments is polar. So actin monomers, which have ATP bound, like to bind to the end of the filament with ATP bound monomers. And once a monomer has bound, then the ATPase activity of actin starts, hydrolyzes ATP to ADP, and then we get an ADP end. And the affinity of the ADP end for monomers is much smaller than the affinity of the ATP end. And thus we get a filament mainly polymerizing at the ATP end and preferentially depolymerizing at the ATP end. And in this way, you can imagine such a filament lying on the table. You can move the whole filament by directed polymerization without moving any of its monomers. Yeah? So this is a clever way of generating motion, and this is in the core, in the center of all this actin-generated motion. So we have the cell with this flat protrusion, and polymerization is going on here at the very tip, and it generates a network assembly rate. And part of this assembly rate is turned into protrusion, and the other part is turned into network retrograde flow inside the cell. And this flow experiences frictions with structures which are stationary in the lab frame of reference. And these are mainly adhesion structures tying the cell to the substrate. And this is in many cases these integrin receptors binding to fibronectin on the substrate surface. So this is essentially the process or the core process which makes those cells here move. So the other one moves too. No, okay. So this is what's going on in the protrusions and what generates motion essentially. So constitutive relations. What are constitutive relations? So they are very popular in physics, and in physics they define the properties of an object you are investigating. For instance, a piece of solid state body. Here we see as an example the relation between current and voltage of two different objects. One has this linear relation, and this linear relation tells us that the resistance of this object is constant, and the mechanism of conduction is most likely that an electric field inside the body causes a drift of free electrons. So this current voltage relation looks quite different, and it is a characteristic of a diode. And this shape tells us that there is an interface between a p-type semiconductor and an n-type semiconductor generating depletion zone of electrons, and this is the conduction mechanism. So we don't want to talk about diodes. The message is the type of relation, the type of the constitutive relation carries information on the mechanisms causing it. It's not the specific values, but the shape and type of relation carries this information. And constitutive relations are also used in medicine, for instance, the pressure volume loop of the heart. And the questions we would like to follow up is, do they exist in cell motility? And as I told you, so constitutive equations, relations are a quantitative definition of an object. So this question is analogous to the question, can we find a quantitative definition of cell motility? And then we would also like to know to which mechanisms these constitutive relations are related. So these questions will guide us through the talk. Before we go on, I need to introduce a term, and this is persistence time. So cells are active walkers changing their direction randomly. And the time passing between two turns is the persistence time. So the time a cell keeps its direction is the persistence time. Here you see the trajectory of a mouse fibroblast recorded on a two-dimensional flat substrate, and you can see it pretty much looks like a random walk. So the cellular properties shaping this trajectory are defined by the relation between speed and persistence time, and this describes the turning behavior. Then they are also characterized by the speed adhesion relation, describing the effect of adhesion density on the velocity. And in some cases, the stationary force-velocity relation, which described the effect of an external force or mechanical resistance to cell velocity, also is relevant for the shape of these trajectories. So we suggest these relations to be constitutive relations of cell motility. So everything which moves according to these three relations moves like a cell. This is essentially the concept of constitutive relations. And we can apply this concept if we can find some general properties of these relations across cell types and conditions. Good. So talking about the relation between cell speed and persistence time. Mayuri et al. Have suggested in 2015 that it exists. And they have recorded it for many different cell types, and then they'll be able to rescale all relations of the different cell types to such a single curve here. And we will come back later to the shape of this relation. And this idea has been picked up by a few other publications listed here. So ideas on the speed adhesion relation date back even further. So already in the 19th, the Horwitz lab measured the speed adhesion relation with different cell types. And this has been then repeated by several labs and with a variety of cell types. And turned out that all speed adhesion relations are biphasic. So first, with increasing adhesion density, the velocity increases, and then with further increase of adhesion density, the velocity decreases again. So here this points already to a general property. The third constitutive relation is the stationary force velocity relation. So this is a response of a constant of the cell velocity to a constant force. This here is a theoretical prediction. Surprisingly, this relation has not been measured yet. So the dynamic force-velocity relation is a response to an elastic force resisting motion. And this is much more popular and has been measured also by different groups and has also been theoretically described and explained in a study in which Alan Ehrlicher was also one of the co-authors. But unfortunately, it is not a constitutive relation since it's a response to a dynamic situation. So moving on to morphodynamics. So morphodynamics is another of these very general properties of mesenchymal cell motility. Here we see 3 examples of cells exhibiting morphodynamics. So they move and constantly change their shape quite dramatically. And all these 3 guys are on 2-dimensional substrates, but we can also see this on a 1-dimensional substrate. So the motion of this MDA MB231 cell has been reduced to 1 dimension by printing a 1D fibronectin lane on a flat substrate. And it also shows heavily changes of shape, so very intense morphodynamics. And this is a simplification of the whole problem we will pick up. So we reduce cell motility now to 1D motion on fibronectin lanes. And then we will first develop a biophysical theory to identify mechanisms. And then I will present experiment and theory on morphodynamics and the 2 constitutive relations. By the way, if there are any questions, just interrupt me and let me know, so I'm ready to answer questions during the talk. So the biophysical model starts from an idea of a cell which has two protrusions, one at the front and one at the back, and the cell body with the cell nucleus. And then the derivation of the equations starts from the force balances. So at the leading edge membrane, we have the force arising from an elastic interaction between the leading edge membrane and the nucleus, and the drag of all this stuff between leading edge and nucleus, which is moved forward. And this force drives the retrograde flow, so it's equal to the friction force caused by the retrograde flow, or which is required to generate the retrograde flow. And this is similar at the back. Again, the elastic interaction with the cell body, the drag of all the stuff in between, and then the friction force of the F-actin retrograde flow in the back of the cell. And the cell body just responds to the two elastic forces from front and back acting on it, and also it has its own drag resisting cell body motion. So the values of the friction coefficients are controlled by integrin signaling. And integrin is a receptor binding to fibronectin. So they are actually in the end controlled by the fibronectin density on the substrate. And here we have chosen this very simple response function, typical for the response of a signaling pathway to stimulus. So friction coefficients and drag coefficients are controlled by fibronectin density. And then the de-adhesion force at the rear is proportional to the velocity. So the faster the cell moves, the harder it is to pull it off, the adhesion sites at the rear. Then at the front, the polymerization rate has a nonlinear relation to the network extension rate. This goes back to this excellent work by Assam Gulami from 2006. And then we have a nonlinear relation for the friction force of retrograde flow and retrograde flow velocity. And this heavily nonlinear friction force relation results from the clutch mechanism of retrograde flow. So retrograde flow goes on in the protrusions, and the protrusions are adhered to substrate. So we have the adhesion structures inside the cell, and their integrin receptor binds to fibronectin, and fibronectin is connected to the substrate. And these adhesion structures are also connected to the F-actin network flowing retrogradely. Now, when this flows, it stretches the bond, and finally the bond may rupture. And the bond may rupture between fibronectin and integrin, or between fibronectin and the substrate, or between the adhesion structure and the F-actin network at any place. So this type of friction mechanism generates heavily nonlinear relations between the retrograde flow velocity and the friction force. It's a very general phenomenon. So first, the friction force increases because the bonds need to be stretched. But at a certain critical velocity, more bonds rupture than form. And then suddenly all of them break, and we have a jump of the velocity to the viscous branch if the driving force is maintained. And this is a stick-slip transition, very versatile phenomenon in nature, causing earthquakes, making boat string instruments sound and things like that. and this stick-slip phenomenon is called the clutch behavior in the field of cell motility. And this has also been measured, observed, and confirmed by different groups with different cell types in different labs. So it's a very basic observation in the field. So we introduce this stick-slip dynamics or the clutch in our model by the dynamics of the friction coefficients. And the dynamics of the friction coefficient is the dynamics of the number of bonds between the F-actin network and the substrate. Yeah? And then we have here a term describing bond formation. And here we have a term describing the breakage of bonds. And this term here, the rate of breakage is proportional to the exponential function of the retrograde flow velocity. And this goes back to the investigations of dynamic bonds by Evan in the 90s. So the rate with which a bond breaks is exponentially dependent on the rate with which you increase the stretch. And this is what enters here. And then we have a noise term describing the random formation and random breakage of bonds. And then we have one equation for the front friction coefficient and one equation for the back friction coefficient. These equations have been derived in Mogilner's group in 2015. And then what we need to add is the dynamics of the length of the protrusions. And this dynamics is simply the difference of velocity, so front edge velocity minus cell body, and analogous for the back protrusion. So this is the complete model. So it's a very simple model, just for ordinary differential equations. So we will sometimes use a model with one protrusion and sometimes a model with two protrusions. And I indicate this by these symbols on the slides. Using one protrusion has the advantage we can do things analytically and do not need to rely on simulations. Now we come to the experiments we have done. So in order to reduce motion to 1D, we have printed lanes of fibronectin on flat two-dimensional substrates, and we varied the fibronectin density, as you can see here, from the different intensities of fibronectin fluorescence in the lanes. And you can nicely see the MDA and B231 cells reverse directions, so we can measure persistence times and speed. And they move across different fibronectin densities, so we can measure the speed-adhesion relation. And cells move with constant length or oscillating length. That means we can observe also the morphodynamics. And we investigated 400 trajectories recorded with a temporal resolution of 30 seconds and 17,000 trajectories with a temporal resolution of 10 minutes. And this large amount of data allowed us also to get very good statistics. So we start with some simple warm-up exercises. When a cell crosses a fibronectin step, so goes from one density to another, the length and the velocity of the cells changes in specific ways. Here you see a chromograph of such a cell crossing steps. And then here you see that the velocity changes and also the length changes at the steps. And then getting statistics of many of those transitions for many trajectories, we can simulate the population average values, and you see we can obtain pretty good agreement between theory and experiments for the different cases of steps and motion on homogeneous lanes. So this is initial parametrization, but it also confirms our choice of force between leading edge of the cell and nucleus. We have chosen an elastic force for this interaction, and this choice is confirmed by these experiments. And here two more warm-up exercises. Here we now observe how cells move across fibronectin bumps of a height of 10 nanogram per square centimeter. And this is recorded by looking at a range of 40 micrometer length. And this range contains a bump. And if the cell leaves the range in the same direction as it has entered it, it was a pass. If the cell leaves it in the opposite direction, it has entered it was a direction reversal. And if it did not leave the range within 5 hours and 30 minutes, it got stuck. So these are the three cases which might occur. And here is a comparison of simulations and experiments. And we could obtain pretty good agreement between theory and experiments. So here now we look at the behavior at steps. And the first panel shows data averaged over a range of initial concentrations, so the concentrations where the cell starts. And then we plot it here in relation to the step height. So also with steps going down. And again, agreement between theory and experiment is satisfying. And here we show the same data, but now for individual pairs between these two densities. So these things are first confirmation for the model, first for parametrization, but we did not go for complete agreement of every detail because the parametrization was complete after we had fitted all the data I will present. Yeah? So this is what we tried to capture as many data as possible with a single set of parameters. And this is why agreement is not always perfect. Let's move on to morphodynamics. So okay, so there are raised hands, but I cannot see them. So are there questions?
Hi, can you hear me? I have a question,
Yes,
please.
yes, I can hear you.
Thank you. I'll also send it in the chat. So my question is for the length dynamics at fibronectin steps and parameterization. So the question is that how does the elastic front-back interaction assumption influence predictions for cell behavior in environments with more complex ECM patterns beyond simple FN steps?
This, I think, as long as the processes are slower than the equilibration of tension within the cell, which takes for the affecting network inside the lamellibodium takes a few seconds. I would assume, based on these data, that also if you would apply more complicated structures here, the elastic force model would still work pretty well. Yeah, so that's a question of time scales. The affecting network is viscoelastic, so if the time scale of this viscoelasticity is a few seconds, so as long as processes, adaptation processes and the geometry are slower, this should work. This would be my guess.
I got it.
Yeah. So we moved on to morphodynamics. And here you see a cumograph of a cell recorded for 48 hours here. And then we observed 4 different dynamic states. So we see cells which are just spread, which do not move, and they have a steady length. And then we see cells which are spread, but they have an oscillatory length, so their length oscillates. And we find cells which move with a steady length, and we find cells which move with an oscillatory length. So these 4 types of morphodynamic states we have observed, and all 4 of them have front and back protrusions. This is a reason why we work with a 2 protrusion model. Interestingly, we do observe apparently spontaneous transitions between these dynamic states. So here between going down and going up, and here between other different dynamic states. And we did not apply any perturbation, and we could not identify any perturbation on the fibronectin lanes causing these transitions. So they are obviously spontaneously, but this means that these cells are multi-stable. So for a given experimental condition, they can be in either of these dynamic states and can switch between those states. So now let's apply our model. And up here you see a comparison between experiment and theory. We find the same set of dynamic states in the model. And sometimes we observe these very smooth oscillations, and you can see we can reach pretty good agreement with regard to amplitudes and theory. And this also applies to the moving, steady, and oscillatory states. So here we compare oscillations in more detail. So you can see that we reach also pretty good agreement with regard to oscillation amplitudes and periods, or velocity amplitudes and periods, and the same applies to front lengths and the length of the back protrusion. And these oscillations are caused by the slip mechanism of the noisy clutch. This is in the center of the oscillations. So it turns out that the steady states are excitable, at least in parts of their parameter regimes. So when we simulate them deterministically, they are steady, but when we switch on the noise in the friction coefficient, they very much look like the oscillatory states, both for the spread state and the moving state. So this is indicates excitable dynamics. So what about the multistability in the model? So we also find it, as shown here in a bifurcation diagram, relating the value of the friction coefficient at different states to the fibronectin density. So at very low density, we only have the spread steady state, but then at a branch point, the 2 moving states bifurcate, and the spread state still exists. And here we have a coexistence of MS and with SS, but then Hopf bifurcations occur and oscillations start, and then we get coexistencies between different dynamic states. And if you changed parameters of the system only a little, then the order of Hopf bifurcations changes with increasing fibronectin density, and you get more different pairings of coexisting states. The moving states exist as state moving up or state moving down. And this is not trivial. So this is not just a consequence of symmetry, because we see transitions between these states. So we see how cells spontaneously change the direction. So these are really two different and discrete states of cells. So within the paradigm of our theoretical network, the state transitions are caused by noise. And that means that the state fraction statistics in the population of cell and the state transition rate statistics are the verification of the theoretical choices for the noise. So now we need to do population simulations for populations of cells, and we do so by identifying a reference parameter set. And then we simulate a cell population by sampling all the cells within a plus minus 5% range of the parameter values of a reference parameter set. And then we obtain the results shown here. So the left-hand side shows the state fractions in the population of cells, and the right-hand side shows the transition statistics. So the state fractions agree pretty well between theory and experiment. And over here, we see all possible transitions between the dynamic states, the morphodynamic states, and the full lines indicate transitions between dynamic states of the deterministic model, of the noise-free models. And the dashed lines indicate state transitions which you observe in the data, so both the experimental and simulated data, but they arise from the fact that a cell which is in the excitable steady state at some point may turn randomly by noise into the in the oscillatory moving state, and similarly for the other pairings. So here you now see the comparison of the statistics of transitions for experiment and simulations. So the transitions occur from the state in the lower row to the state in the upper row. And again, we can obtain satisfying agreement between theory and experiments. And our noise choices for the noise in the friction coefficient has been simply the rules for chemical mass equations. Yeah. So this the square root of the sum of rates divided by a normalizing factor. So we can repeat this analysis, but now with applying latrunculin to the MDA-MB231 cells. And here we see the state fraction results. And again, agreement between theory and experiment is satisfying. Latrunculin reduces the polymerization rate, so the network extension rate. We can also apply blebistatin. Blebistatin decreases contractile forces inside cells by contraction by myosin motors. And in the end, this reduces the stress fiber density and the adhesion site density inside cells. So the density of structures with which the radiogate flow experiences friction. And also for this case, we reach pretty good agreement between theory and experiment. So now let's move on to the constitutive relations of cell motility, and we start with the speed adhesion relation. And we do so for a model cell with a single protrusion, and then we can write down the speed adhesion relation analytically. So this first equation here relates the cell velocity to all the parameters of the dynamics. W0 is the Lambert V0 function. And this is the dependency of the coefficients on fibronectin density as seen before. And here we allowed for two more forces. So Fc might be a contractile force from myosin acting inside the cell on the F-actin network. And Fm is a force which also might be an external force acting on the leading edge membrane or maybe pulling from the back. Here we have only one protrusion. And both of them do not depend on velocity. So both of them are constant when you change velocity. So then we can fit this equation to our MDA MB231 data. And this is the result of the fit shown here. And interestingly, both velocity independent forces turn out to be zero. This comes out of the fit. And then this means we obtain this expression here for the speed adhesion relation of MDA MB231 cells. But then we can go on and fit this equation also to earlier published data for different cell types, here for CHO, keratocytes, and PTK1 cells. And we can fit all of them rather well. For keratocytes and PTK1 cells, we also had data for retrograde flow, which we could include in the fits. And all of them can be described by this equation with a pretty good agreement. This here is a compilation of several adhesion velocity relations done by the Horwitz lab. But here we had too few data points to really get a reliable fit. So this is not a unique fit here. And in all cases, again, the velocity independent forces turned out to be zero. So the different shapes of the speed adhesion velocity relation result from the different dependencies of the friction coefficient and the drag coefficient on fibronectin densities coming out of the fits. So here you see these different dependencies for the 4 different cell types. And then this explains the different shapes of the force velocity relations we observe. But as a rule of thumb, we always find that the larger friction relative to drag, the faster is the velocity. And then we can calculate also things we cannot directly measure, for instance, the network extension rate or retrograde flow velocity. And then in all cases, it applies that when you increase the fibronectin density, the friction coefficient increases first, which generates larger friction force driving the front protrusion. And this causes the rising branch of the speed adhesion relation. And then the drag coefficient follows at slightly higher adhesion density. And this pulls from the back and causes a falling branch of the biphasic speed adhesion relation. So now let's come to the second constitutive relations of cell motility, and this is the speed persistence time relation. And this relation is the statistics of direction reversal state transitions. So if you want to understand this relation, we need to understand the statistics of direction reversals. So here you see a simulation of the scenario of a direction reversal. In the upper panel, you see simulations. In the lower panel, you see experiment. And the curves which are shown are averages over many, many trajectories plotted in the background here. So what we see is that first, the back velocity slows down. And it becomes slower and slower. And the time of direction reversal is defined as a time when the cell nucleus changes its direction of motion. So even before the nucleus changes its direction of motion, the back edge starts to move in the opposite direction with negative velocity. But when the nucleus starts to move in the opposite direction, the front still moves in the same direction, but the back keeps pulling, and finally the front collapses with a large negative velocity peak, and then both of them settle into motion in the new direction. And the scenario coming out of the experiments is very similar. And you find more quantitative comparisons of directly measurable quantities in this publication here. So the scenarios of theory and experiments agree, and this encourages us to go on. And in these simulations here, we investigate the mechanism of direction reversal with a deterministic noise-free model in a little bit more detail. And we do so by applying a perturbation to the back friction coefficient. And we do so in the subcritical case and in the supercritical case. So we start with a subcritical case. Here we perturb the drag friction coefficient, and immediately the retrograde flow in the back goes down. And this causes the protrusion, because polymerization goes on, the protrusion at the back to move in the opposite direction, and it starts to pull. But finally, it is not strong enough, and the force is shown here. And what happens is that the front protrusion wins, and the back protrusion collapses with a large peak in the retrograde flow. And here you can see this in experimental records. So we have two cells moving from right to left, and you can see here that the back protrusion starts to become active, and it starts to pull, but it's just not able to stall the front protrusion, and the cells moves in the old direction, goes on in the old direction. And the same even here, with a more impressive case here, here the front protrusion appears to vanish, but finally it wins. So in the supercritical case, we perturb the friction coefficient to a value larger than the friction coefficient at the front. And then in the end, the back protrusion collapses, and the cell changes direction. And this you can see here in this movie of a cell changing the direction several times. So we learn from the simulation, direction reversal occurs if the friction coefficient in the back becomes larger than the friction coefficient at the front. So interestingly, the force collapsing the front protrusion here has a value in these simulations very similar to the stall force of the protrusions of fish coracocytes measured in this study here 13 years earlier. So something is special to this value, which we have not completely understood yet. So here you finally now see the speed persistence relation for 4 different cell types, and it turns out that it's also biphasic. So with increasing velocity, the persistence time first increases and then it decreases again. And the speed which we measure here is simply the average speed for this phase of motion between 2 turning points. So the speed persistence relation is biphasic for all the 4 cell types we have investigated, and it is so in experiment and simulations. And published data show only the rising branch up to here, and the data I have shown you initially from this Mayuri et al. Publication are all in the ranges of these rising branches here. So the biphasic character of this relation is even maintained if we apply latrunculin or blebbistatin, so the drugs we have used before. And especially the application of latrunculin is important for a discussion on the mechanism causing the speed-resistance relation in the field. Here, the application of latrunculin increases persistence times, and it does so both in experiment and simulations. So the speed-persistence relation is biphasic. How can we explain this? So from the deterministic simulations I have shown you 2 slides earlier, it is clear that a protrusion is, or the persistence, the polarization of the cell is more stable the larger the difference between front and back friction coefficients. And here you see when we increase the speed, this difference increases. So with increasing speed, the polarization becomes more stable, and this causes a rising phase of the speed-persistence relation. And then when the cell speed increases, also rate-to-gate flow rate increases, and this causes an increase of the noise amplitude in the friction coefficient at high velocities. And finally, this increasing noise amplitude, which also allows for transient occurrence of larger friction coefficient values, then causes a falling branch of the speed persistence relations. So then we can look at the speed persistence relation at variable densities of fibronectin and compare experiment and simulation again. And here we see that with increasing fibronectin density, we get more persistent behavior, both in experiments and simulations. And this can be explained by increasing differences between the friction coefficient, which is shown here, and this dependency can also be explained then by the noise amplitude. So we see that at very high fibronectin density, these 2 curves below here, we have a small noise amplitude, and this causes long persistence times. And at smaller fibronectin density, we have larger noise amplitudes, and this causes smaller persistence times. An interesting observation was that the falling branch of the speed persistence relation is lacking at very high densities. Yeah? And this is due to the speed adhesion relation in the end, because it tells us that the high velocities required for the falling branch do not occur anymore at these high fibronectin densities. And this also occurs in theory and simulations. So this is what I wanted to tell you. And I'd like to sum it up here. So cells exhibit morphodynamics, the speed persistence relation and the speed adhesion relation simultaneously. So while they move and constantly change their shape, they obey these 2 constitutive relations. And we suggest that both of these relations, speed persistence and speed adhesion, can indeed be perceived as constitutive relations of mesenchymal cell motility. So that means cells are things which move according to these relations, or things which move according to these relations move like cells. Yeah? And then both relations are biphasic. The morphodynamics is multi-stable. And this is also a versatile observation, similar versatile as morphodynamics on its own. And then a biophysical theory which starts from very simple observations. So the clutch mechanism of rate-to-gate flow the force balances and integrin signaling can explain all of these observations. And here I'm pretty sure if this theory wouldn't be simple, it couldn't explain this variety of observations. If it would have very specific requirements, it could not be as versatile as it is. And in the end, this suggests a picture of cells that they behave as nonlinear, multi-stable, mechanical dynamical systems. And the parameters of this dynamical system are controlled by signaling, integrin signal, GTPases, and so on. Thank you for your attention.
Thank you very much. Questions? Go ahead, you can unmute yourself and ask. This is not my area, otherwise I would normally have one or two questions myself. Quite far away from my area.
Yeah.
But while we are waiting, let me ask a very general question, if I may, really while we are waiting. So I'm very curious about this notion of constitutive relations. If I understand this, so I'm familiar with the sort of, these are essentially phenomenological laws, right, at a higher level that hopefully
Yes.
will also constrain the underlying mechanisms in a way that they fall into one of these classes. So in other words, modeling these sort of characteristic equations for a system teaches you something about the underlying structure. Is that the goal of how we think about this?
Exactly. Exactly. So this is one way of using them. Yeah. So if you see them, you can draw conclusions on the mechanism. But also, if you want to design a model of a multi-cells, which addresses questions not directly related to the constitutive equations. Yeah. But then you first need to develop a model which obeys the constitutive relations, and then you can go on. Yeah. Because once that model obeys the constitutive relations, it is a model of cell motility. Yeah.
Yeah.
So this is how you can use them. Yeah.
Is there because this pattern shows up in other places, is there a setup where multiple structural setups all lead to similar constitutive relations? In other words, many paths to a final for unknown reasons or known reasons, that you see certain patterns evolve again and come up again and again, even though the structure is not apparently similar. Is that making any sense, what I'm asking?
Now, so what we have done is if I understood your question correctly, it is, do constitutive relations show up in many different
like a universal? Yeah, exactly.
Yes, exactly. So this is the whole idea. Yeah, that as long as you have, in this case here, mesenchymal cell motility, you should see the constitutive relations. Yeah? And we see it in control situations, in lanes fabricated by microcontact printing, on lanes fabricated by photo patterning, with latrunculin applied, and with blebistatin applied. So this is a set of experimental conditions we have investigated, and 4 different cell types. Yeah. And this Mayuri et al. Publication earlier on has observed this rising branch for about 40 cell types, and in 1D, in 2D, and in 3D motion. 3D then is on fibers of the extracellular matrix. No, through networks of fibers of extracellular.
Right.
So this is a whole idea that you really see it in many different experimental conditions.
So across fields, sort of, is there sort of a attempts to build a theory that tries to explain why all these different paths lead to these common outcomes that can be captured with these simpler phenomenological equations? So is there a theory, say in physics or in general, like that, where successes have been made, right? There, because if you try to model it at the micro level, you would find that the structures are very different. That's what you were trying to say, right? Also with the different methods. So why is it some kind of evolutionary argument that they all end up obeying certain properties, that the ones that we see around us are the ones that somehow are filtered to observe these phenomenological relations? Again, I don't know if my question makes sense.
The question makes absolutely sense. So I would not like to answer it with an evolutionary argument, but rather, so somehow here it is, okay. So I'd like to stay within our framework, yeah, and this framework uses only very general observations, yeah. So we simply use the force balances, and what we also use is this clutch mechanism of retrograde flow, yeah, this. And this is really a very general phenomenon. Yeah. So this is friction due to transient bonds. And for instance, if musicians put colophon on the bows for string bows instruments, this is in order to generate a transient bond between the strings and the bow. Yeah. And then you can generate this nonlinear dynamics, and it causes earthquakes, it causes certain diseases in joints and things like that. So this is a very simple way of generating nonlinear behavior. So that means all biological structures which generate friction by transient bonds, which can be a large variety, yeah, are able to generate this friction law.
I see.
And in this case here, this is probably the reason for the versatility of the observations.
Yes.
Yeah.
Great. I see a raised hand, but that's you. So if there are no more questions, I guess we've reached the hour as well, 5 minutes past the hour. So thank you very much, Martin, for this great talk. And we'll have the recording up as well. So if people have questions, I guess they can reach out to you later as well. Thank you very much for this.
Thank you for listening.